Hi guys, so this is a video exploring the synoptic links between the ideas that we covered in Module 2, Lesson 16 to Lesson 20. So that's looking at the non-specific immune response, the specific immune response, and how things like monoclonal antibodies and the ELISA test and things like that start to link into all of those ideas. Being able to link the ideas is really key. As you know, we've got paper 3 next year, and the idea is that you should be able to start drawing together these more complicated concepts to view a whole system. So I'm going to be writing in a few different colours you can see down the side. I'm hopeful you can see them all. If not, I apologise. And if you've got any feedback about the colours and things like that, please just let me know and I'll change it for next time. Okay, so we're going to start by looking at the very beginning, and we're going to start with lesson 2.16, the non-specific immune response. And this is going to be our starting point because this is how every single response begins, and this is our first line of defence. So when we're considering our first line of defence, we know that the idea there is that we are responding to pathogens. And pathogens is one of our keywords. We've got to be able to recognise it. I'm just going to underline that for you. Pathogens, as a definition, are organisms with the ability to cause harm. So foreign or non-self microorganisms that cause harm. Okay, and now these have got non-self antigens on their surface. And again, you'll see in lots of instances in exam questions where it asks, what are antigens? Okay, and you have to be able to define them. So definition-wise, these are proteins on the cell surface membrane. I'm just going to abbreviate it to CSM. If you're not sure, cell surface membrane, that can be recognised as self or non-self and work to trigger the immune response. And the idea there is that these antigens are there to kickstart everything. So we have to say, as the key part of the definition, the bit that will be underlined on a mark scheme, they trigger the immune response. They are there to cause some kind of physiological change. Okay, so we've got those pathogens, so we've got non-self antigens on the surface. Our pathogens are going to end up using their antigens, and I'm going to abbreviate antigens to AG, and they're going to bind to non-self receptors, okay, because they're not our body cells, on some kind of body cell. Okay, and if you can't remember what a receptor is, if you think back to the beginning of topic 2, these are proteins, normally a form of glycoprotein, and they are normally extrinsic, so they're protruding out of the cell, that are complementary to the antigens. Okay, complementary to another molecule, but in this case we are talking about those antigens binding to them, to the antigens. Okay, so now we've got that binding, we've got a little bit of a cellular response to begin with. So as you can see, I'm moving down the page, and hopefully it should be nice and big at the end of this video. So if you need to pause at any time, pause. I'll try and keep the previous point visible in the screen as well, just so you can see where those ideas link. Okay, so once we've got that antigen binding to non-self receptors on that body cell, the body's going to be like, oh hell, I've got something inside of me that shouldn't be. And what we've got happening then is we start our non-specific immune response. Now in this, we are going to be relying on our white blood cells, and we can name an example for this one. We can say that we're going to use some macrophages, and they are going to engulf. And engulf is a key word to be able to use. It implies that the phagocyte is going to be able to wrap itself around and take in that pathogen. Okay, a bit like if you look at the diagram quickly, so we've got the macrophage and the pathogen, so macrophage and the pathogen, and what it does is it sends out these little extensions of its body to engulf that pathogen and enclose it. Now this is all well and good, but we need to know how that happens, and we're going to start thinking about that process a little bit more now. So the macrophage is going to engulf the pathogen via our key process, phagocytosis. You've got to be able to spell that word, so if you're not too sure, could you please pause and make sure you know how to spell that word. And so, for phagocytosis, we have got the pathogen being enclosed in a phagosome, or a phagosome, depends on which way you want to say it. But again, these are key words. So we're enclosing it in a phagosome. That is an example of a vesicle. Okay, so it's a membrane-bound organelle, a membrane-bound sac, and it's found in the phagocyte specifically. Okay, so specific location. What we've got once we've got our pathogen enclosed in a vesicle is that we fuse it, or fuse that phagosome with a lysosome, and that contains lysozymes. Okay, now lysozymes, we should know, are hydrolytic enzymes, so they're going to help us break stuff down through hydrolysis reactions. Hydrolytic enzymes break down molecules via hydrolysis. So that is to say that if we're thinking about things like our phospholipid bilayer, for example, as soon as we start undergoing hydrolysis, it's going to start breaking down those phospholipids into the phosphate head and the fatty acid tails, and maybe even break down those fatty acid tails further. Okay, we don't need to know too much about the whole process of hydrolysis in these membranes, we just need to know that they occur. The fusion of the phagosome with the lysosome forms that big word that we hate writing, but we need to be able to, phagolysosome. And those hydrolytic enzymes are going to work together to break down the pathogen. And that breakdown of the pathogen is key because everything follows on from there. We know that if we sort of go back to looking at a quick pass, the macrophage is going to engulf the pathogen via phagocytosis. That results in the breakdown of the pathogen inside the cell. And we get the antigens from the pathogen being displayed on the surface. So pathogens, antigens are displayed on macrophage surface. And that, we know, is called an antigen presenting cell. And for abbreviation's sake, I'm going to write APC from here on in. Okay, so that's as far process-wise as we need to think about that first line of defence. When we're considering that first line of defence, it only occurs after the physical barriers have been breached. Okay, so it's just important to remember that the physical barriers have to be broken, so maybe there's a gap in the skin or something like that, before any of this is going to happen. Okay. So after we've got those antigen presenting cells, we're then able to trigger our specific immune response. And I'm going to move to just subhead that in black now. So we've got the specific immune response. And if you remember looking at lessons 2.17 and slightly moving into 2.18, we've got two different branches of the specific immune response that we can have. And we're going to look at the first example up here in green. Now this is the faster of the two responses. It's going to occur a lot more readily. It means that we're going to trigger the second response, or the second type of the specific immune response, afterwards. And this first type is the cell mediated response. Now this is a quick response. It's specific, as we know, because it's part of the specific immune system, and it is there to fight viral infections. Okay, and by fighting viral infections, we are going to result in cell damage, because remember, those viruses replicate inside our cell, so we need to be able to destroy those cells that are infected. And it all starts off with a T helper cell, which I'm going to abbreviate to TH, so T helper cell, binding to the antigen presenting cell. Okay, so we're going to have a receptor antigen complex, and that triggers off a massive cascade of biological reactions that are going to help us respond to this pathogen. So the T helper cells release those chemicals that we spoke about in lesson 2.17, those interleukins. And remember, we can abbreviate those to ILs, and they are going to trigger mitosis. So we're going to trigger mitosis of T helper cells around them, and we're going to get a lot of clones produced. Okay, they're genetically identical. Remember that from mitosis when we looked at it in this module. You might want to take time now to look back over the lessons for mitosis, because remember, that's still part of module 2. And we've got a few different types of cells that we can produce from these clones anyway. We've got our most dangerous type, those T killer cells. Sometimes they're called cytotoxic T cells, and abbreviation wise they might look like big T and then a small c in an exam, for example. And what these cytotoxic T cells do is they produce perforin. And we know that perforin is going to produce holes in the membrane. It's going to punch holes in our pathogen membrane, in our infected cell. So pathogen or infected cell membrane. And ultimately, this is somewhat useful because it means that the cell is going to die. We're going to destroy that infection and hopefully get rid of any pathogens at the same time. Now, you don't have to know in detail how perforin works, but there is the chance, and we're going to draw links to other modules or links to other lessons, that they might decide in an exam to bring in osmosis here. Because remember, if we've got holes in a membrane, we can start saying there's a difference in water potential and things like that. Okay, we've also got T memory cells. And these are the really important ones in terms of long-term responses to infection. They're going to persist in the blood. And when we encounter the same infection, so if I link that back to the interleukin stuff, so on the second infection, they are going to help trigger the release of interleukins, mitosis, and we're going to get lots and lots of those T cells. But in the first instance, and the key term that you need to be able to use to describe T memory cells is that they are part of the immunological memory. And finally, we've got a final type, a third type of the T cells that we don't really talk about too much, but they're really important when we start looking at the second branch of the specific immune response, and that is the activated T helper cells. So these are T helpers that help another process, but don't necessarily produce those interleukins that are going to cause more T cells to be produced. Now this, for the moment, is all we need for the cell-mediated response. We're going to go back to where we started looking at the specific immune response, going to pick a different colour, and we're then going to take a look at the slightly slower, but equally important humoral response. And in this, we are responding slower, as we know, and we are responding to bacterial infections. So we're going to be looking at how we're going to produce the antibodies that help us fight infection all the time. Now at this point, it's important to remember that B cells can also act as antigen presenting cells. Okay, and while we've got those antigen presenting B cells, they are triggered to divide using those activated T helper cells. So T helper activated triggers B cells to divide by mitosis. And again, we're going to be using those interleukins, those IL molecules. And once we've got those B cells dividing, we've got a process called clonal expansion happening. So clonal expansion, and that is just where we're producing lots of the same B cells. Obviously, we don't just need one cell to cause a response. We're looking to get lots and lots and lots and lots of those responses. Now, we've got, again, a couple of different types of B cells that we can produce, each of which has got a different role. So we've got plasma cells, which are the nicely active ones that help us in the fight against infection. And these are going to produce specific antibodies. Okay, I'm just going to write AB now because I'm frankly lazy. And those antibodies are able to affect our pathogen, affect cells in 3 different ways. So we've got neutralization, we've got agglutination, and we have got opsonization. Okay, and we're going to take each of those in process or in turn quickly and just explain what they mean again. So neutralization is where the antibodies act as antitoxins. And if you're not sure what toxins are, the toxins are produced by bacteria, and those are the things that make us ill. They give us the symptoms that we see. Okay, and once they act as those antitoxins, they are able to stop the effect of toxins, so we don't get as sick. Now, agglutination works sort of alongside opsonization, so that I'm going to link them just there. And agglutination is where your antibodies attach to more than one antigen. And this is normally, these antigens that they attach to are normally found on more than one pathogen. Okay, so that means that what we actually get is a clumping of pathogen cells. Okay, so they're in a big block now rather than just individually and floating around. And the reason why I've linked this to opsonization is because the antibodies can also flag up pathogens for destruction. So they can sort of highlight them to the body, and they can help increase the chances of phagocytosis in that cell type. And obviously, if we've got a bigger clump of pathogen, then that's also going to increase the likelihood of them being visible. Now, if we take a quick look at the whole structure of antibodies, so sticking with lesson 2.17, we know that our antibodies are Y-shaped molecules and are made of these two heavy chains that I'm drawing now that are hopefully going to be equal, and then we've got two slightly lighter chains that bind to them. So we're just going to label those up, heavy chains. Molecularly, these are longer polypeptides. So again, we're bringing in module 1, and the short chains are obviously light chains because they don't have as much mass. And these two chains, the heavy and the light chains, are linked. So we've got the production of a quaternary structured molecule. Okay, because we've got more than one polypeptide chain being bound together to give a functional piece. Now, the reason why it's important to flag these up is because the heavy chains, and specifically the long arms of the heavy chains, the bottom of the Y shape, these bits that I'm highlighting now are the same in every antibody molecule that you're going to get. The bit that differs is the end of those heavy chains and the light chains. And those bits together over here, which I'll flag up, are called the variable region. Okay, so this is the site of antigen binding. And the reason they need to be variable is so that we can bind each antigen to a specific antibody to give us that response. Okay, so remember, with these, we're always talking about specificity. We need that kind of lock and key effect to give us a response that we need. Now, if we go back over to the types of B cells that we can have, we've also got, even more importantly, I would say, again, in terms of memory, B memory cells. And these are like the T memory cells. They're for immunological memory, and they persist in the blood. Now, if they persist in the blood, they're able to move around and effectively visit all of the sites in the body just in case an infection gets in anywhere. Now, when we're talking about the memory cells, we know that we're going to be talking about reinfection, and we've got to be able to recognise how and why the concentration of antibodies in the blood is going to differ depending on their activity. So in lots of exam situations and in lessons as well, you will have seen a graph that looks like this. On the x-axis, maybe we've got time in days, and on the y, we have got antibody levels in milligrams per deciliter. Okay, now the shape of the graph is pretty standard regardless of what the infection is. So we've got an initial point of infection, and our graph is going to show a delay. Okay, so the response and the production of antibodies is not immediate. And this is because realistically, we've got to go through all of this process over here with the dark purple stuff and go through all of the triggering of the B cells to get to this point. Okay, it takes a little bit of time. We've got a slow and short and small increase in the antibody levels. And this is because really our plasma cells are just going to be focused on fighting that primary infection. However, after the infection, my levels of antibody for that particular infection are going to drop off until maybe I get infected again. Maybe I haven't learnt my lesson from last time, and maybe, I don't know, I've licked the same brick for the second time and I've got ill again with the same infection. Now this time we get a rapid increase in antibody levels, and it's a much more great effect. So we've got a big response now. And this is because over time, those memory cells, the B memory cells, are able to very quickly ignore all of that stuff that's happened before, and all they've got to do is trigger the production of plasma cells very quickly. So we don't have to worry about the non-specific immune response and all of that gumph beforehand. We're just going to get those B memory cells to make some plasma cells, and we're going to produce those antibodies. And because we've got lots of those B memory cells, they're going to be able to do that really quickly, and the infection is fought at a much quicker rate. So it's a shorter duration of response as well. So now we can spend some time taking a look at how other ideas interlink with this whole concept, this whole process. Okay, so the immune system currently looks a little bit like this in terms of topic, but we know that there's much more detail that we can add. So the first thing we're going to do is we're going to look at where the different types of immunity fit into this whole scheme of work. So I'm going to pick a nice hot pink colour, and we're going to start up at the beginning and take a look at the natural and artificial active immunities. And they both occur at this very first point. Okay, so we're going to have a couple of branches come off here. We've got our natural active and artificial active immunity happening here. Natural active, artificial active. And the natural active, if we remember, is when your environmental pathogens are going to trigger a response. So this is how we respond to things. If you've got a cold at the moment, that's how your body is fighting it. And again, that artificial active is where pathogens from a non-bodily source, so non-bodily or foreign pathogens, are going to trigger that response. And this is where we get the activity of a vaccine. Okay, so the vaccine works to basically start the process of the immune response to end up with the following products. So we're going to want some T memory cells, product of vaccines, and we're also going to want some B memory cells, so I'll highlight that again as well down here. Okay, so the idea of that artificial active response is that we're getting those memory cells ready to quickly fight any infection if we actually get it. Now remember, if we think about those vaccines again, we're working on the idea of herd immunity, where the few are protected by the majority, or the few are protected by the vaccinated majority. And those few are going to be those that are maybe older or very young, or immunocompromised. So maybe they've had an organ transplant, for example. Compromised. Okay. Then we're going to take a look at where those natural and artificial passive responses are going to come in. So natural passive responses are those where we're just going to get antibodies in our body from a source that isn't you. Okay, so natural, passive. And as we saw in the lesson, I think it was 2.19, we've got antibodies in the blood from the mother. Okay, and these again are transferred either in utero, so in the uterus, or postpartum, so in breast milk. Okay, so the body doesn't have to make them, but they do work. And then finally, in the same vein, again with those plasma cells, we have got our artificial passive responses as well. And these are where we're going to get antibodies from non-human sources helping us. Okay, so the example that we've seen before is tetanus, which is derived from horses, and they are just injected into us if we've come into contact with anything that might have tetanus in there. And the idea of these artificial passive treatments is that they are quick and life-saving responses. And while we're here, we're going to spend a bit of time looking at those monoclonal antibodies. So that again is linked to where we've got these plasma cell bits. So I'm just going to draw a nice blue line now. We're going to use dark blue. And these are our monoclonal antibodies. I'm going to abbreviate that to MABs because, again, I am lazy. Now, we're not going to go into a great deal of detail about how they're used. You can look over the monoclonal antibodies, lesson 2.18, if you're not sure, but they are lab produced and they are used therapeutically. By therapeutically, I mean for testing or treatment. And we make them in the following way. So we're going to inject our mouse with the pathogen or the antigen that we want to fight. Okay, the thing that we're targeting. Once we've done that, we are going to remove the spleen or remove some of the spleen cells, and we're going to combine that with a cancer cell, and that's going to produce the hybridoma. Okay, now that hybridoma is going to rapidly divide and in doing so, lots and lots of antibody producing plasma cell. And the final thing we can do is we can purify those antibodies and use them. Now this is really important because we've said that we're going to use them for testing and treatment. Now testing wise, you should remember from lesson 2.18 that they're involved in pregnancy testing, and they're also used in the ELISA test or the ELISA test. And for treatment, there is lots and lots of scope for the exam board to ask you lots and lots of questions about these. Okay, but the example that they ask you to learn about in preparation for the exam is using them in cancer treatments, and that is for either flagging up the cancer cells, so using them to opsonize, or to deliver chemotherapies. Okay, now we're going to spend the final bit of the lesson talking about what the ELISA test actually tests for, and that means that we are going to be talking about HIV. Okay, now for HIV, we need to recap the fact that we're actually looking over at the impact on T helper cells. Okay, so really, we're looking at where I've put those red brackets. And the way that HIV works is by effectively knocking out, I'm going to put KO, of the T helper cells. It does that by infecting them and using the T helper cells to make more HIV. Now remember, HIV is called a lentivirus. It has a variable latency period. Okay, so that means it can just sit in your body getting worse and worse and worse over time. It's variable latency period. Now, if you're not sure about the structure of HIV, you need to go back and look at the lesson. It's lesson 2.20. But structurally, there are a few key things that we need to remember, that our HIV has got 2 times RNA strands. Okay, so they're single-stranded genetic material. And it's also got 2 enzymes in it. And those are reverse transcriptase and they are integrase. And the way that HIV works, and if you're not sure about the whole process, then please go back to the lesson and take a look at it, is that HIV inserts its genetic material, so its RNA, into the host, and we know that that's the T helper cell, and it uses the reverse transcriptase enzyme to turn its RNA into DNA. So we can make one strand using reverse transcriptase and the second strand using the cell's machinery. So that is to say that we're using the free nucleotides that are floating around in the cytoplasm. So once we've got that reverse transcriptase enzyme taking the RNA to DNA, so I'll just draw little arrows there just so we can see where we're going. Integrase is then going to integrate the HIV DNA now into the host. And what this does is it means that now the host produces proviral molecules. Okay, and the proviral molecules means that these are able to produce a virus or able to build a virus. Okay, so these are bad things to be making. The host produces proviral molecules and then they're used to build new viruses. And the way that our HIV molecules are going to leave the cell is through budding. So this is effectively where the HIV molecules are going to move to the cell surface, so they're going to move to the cell surface membrane, and they're going to be released via exocytosis. And in being released, they take some of the host membrane with them. And this is the bit that makes HIV so difficult to treat, because in stealing the host membrane, it steals some of the host glycoproteins as well. And this makes up their viral envelope, the outside, and a bit like if you put a disguise on, it means they're hidden from the immune system. And this is why they're able to get away with having such a long latency y period. Okay, because we don't know they're there. Remember, they're viruses, so they sit inside the cell, and those, as we've said, are tested for using the ELISA test. Now, hopefully, your mind map should be quite big, quite juicy looking like this. This is only one part of Module 2. Okay, so there's lots more detail that you could add if you want to. You could start over here with the monoclonal antibodies and explain how the pregnancy testing and the ELISA test actually works. You could add a little bit more detail about the whole process of vaccines and how they work to produce the B and T memory cells. There's lots of scope for you to add your extra detail. This is just a basic overview of the whole of lessons 2.16 to 2.20, and I hope you found it useful. If you've got anything else that you'd like to cover, let me know, and I can try and make one of these for you as well. Okay, thanks guys.